Now, while I remember, someone will have to send me an email to remind me to do this for you. Since they were talking about board reviews, I created condensed study guides for all the different physiologies that are part of the boards, except for neuro, since I don't teach it, that I put on there's a site for people who don't pass the ARES, and then they go on, and I put that, and plus some practice quizzes. But I have condensed study guides for all the parts of physio boards except for neurophys that I have. They're PDF files that I can email you if you want. Okay? So I went and I sit there and I look, well, what I think is, remember, I may not put everything on, I don't know, but I think these are kind of important. I took pictures. So there has a lot of copyrighted pictures. I just went on the internet and said, which is the best picture, and stick it on there. So I'll have it and I'll email it to you. So I got it for cardiovascular, for cardiac, I've got it for vascular, I've got it for exercise, phys, reproductive. I kind of broke it into sections. So remember, the only part I don't have is neuro. Okay? Okay, so just someone has to send me an email to remind me, because I'll forget. I've got them somewhere, some file somewhere in my house. I don't keep it here anymore, I don't think. Okay, so what we're going to do today is we're going to talk about regulation of extracellular fluid volume, the role of the kidneys in it. We're going to look at when conditions you want to have a dilute urine, lots of dilute urine, conditions where you're going to have a little bit of urine but it's very concentrated. At the end, we'll talk about what is all over. Oh, this is why you're not supposed to have stuff in the room, because I know. But it's not me. That's the reason why, because that happens. Okay. Hope it's not on your computer. It's on the power supply. Be careful. Okay. Smells good? Oh, yeah, it does. Okay. I had to move towards it. We'll also talk about regulation of osmolality, because it's all linked, osmolality and fluid volumes and all that. So we'll do a lot of this. So we have to cover this material, because I've already written your exam. I already did. Done. Been there, done that. Okay. A lot of the stuff that we talk about, I've already discussed most of it, believe it or not. So we're going to pull out the little chart, this thing, and we're going to draw on it again and use it as a means to kind of go over what we're doing now. So we've really done the hard part. We'll just kind of go through it again. I'll review some other stuff. Okay. So I have the little pictures up here in the front. Okay. So also, while I remember, I'm getting just lazy in my old age and stuff. Is the next exam, it's 50 questions and it's all on Scantron. No fill in the blank. I just got what? Are you mad or are you happy? Okay. I don't know. Some people, but it's just 50 questions. Duh, whatever. Okay. It's not that bad, but some of the stuff's harder. Okay. But I know Jennifer hates renal phys, so we got to make sure she gets it. Okay. So we're going to make sure you get it. Okay. A lot of people, renal phys is hard anyway. All right. Yeah, Lauren too? Okay. So, things to consider before we talk about the scenarios is if all parts of the renal tubule were freely permeable to water, we would have a problem. If it was freely permeable water, your urine would always have the same osmolarity, same volume. Because if all parts of the nephron tubule were freely permeable to water, every time you reabsorb sodium, who's going to come along? Water. And so the osmolarity is never always going to be the same, be boring. So different parts of the nephron tubule, you have to separate water and solutes. So that's where it's going to be important to know different parts of the nephron tubule, which are permeable to water, which are not, and maybe also ones that are permeable to water under certain conditions. Okay, and ADH is going to be a very important factor, because ADH affects water permeability but does not influence solutes, whether or not we reabsorb solutes, but it influences water permeability. So if I have this little generic picture of the nephron tubule, which parts of the nephron tubule, I'm going to use red, what I would highlight to say these are parts of the nephron tubule that have receptors for antidiuretic hormone, and its water permeability depends upon the presence of that hormone. Where would I begin? What part? You need to know your names of the different parts of the tubule. The late distal tubule. So I'm going to say start around here. So here, and then you got, what's this part right there? The cortical collecting tubule. And then here, these are the medullary ducts, where this one was actually distinguishing between an outer and an inner, but the medullary ducts too. So these ones, the water permeability depends upon the presence of ADH, okay? So if you look here, you'll notice in the absence of ADH, presence of ADH, we have a little difference. Same thing's going to be going on along here. We'll review what's going on in that loop of Henle. But look at, when you got ADH around, water is leaving. And where's it going to? It's leaving the nephron tubule and eventually going where? Back into your, it's going, well, it better go back and it's going to the interstitium and then into your blood. You're going to hold on to that water. And so the urine that you excrete with ADHs around, you're going to have a little, it's going to be a small amount, it's going to be very concentrated. The concentration of solutes is going to be greater. If you've got no ADH, large volume of urine, it's going to be dilute. Now, these little dots, what is, I mean, those dots could represent a number of different solutes, but what is the major solute that I will pick up in urine? Urea. That is going to be the primary, the main one. But urea is one of them. You'll have, what's another waste product that you're going to see in urine? It's a waste product. Creatinine. Okay? So remember, creatinine we filter, and you don't absorb it at all. Unlike urea, we do absorb. But remember, we're going to use that for our advantage. Okay? So ADH plays a very important role in this whole process. So in your notes, you notice, if you look at your notes are complete. Did you notice that? Things are written in bold. So anything I have a blank thing, you've got it written down. Oh. So you don't have to, you still have to pay somewhat attention, but you don't have to go, you know, write things down frantically. So, but everything's there for you. So we just went over this. So high ADH, water reabsorption, is that up or down? If you've got lots of anti-P hormone, the water reabsorption, up. Urine volume, down. Low ADH, water reabsorption, down. Urine volume is up. Okay, so scenarios. What could be going on that I would have lots of ADH around? What would be a scenario like, oh, need a lot of ADH. I need to conserve my water. If you're dehydrated, maybe you're lost out in the desert. You don't have ready access to water. You haven't had haven't had water in a while, so your urine becomes a little bit more concentrated. What would be the other scenario? You just drank a whole bunch of, oh, no, sorry, this scenario over here, the low ADH. Well, alcohol, okay, yeah. Go right there. I was hoping just drink lots of water, but okay, we'll go there too. But drink lots of alcohol. That would lower. So, yeah, well, you're at the point I need some alcohol. Okay. Yeah, you know, someone said something to me in class, or swear to God, I thought they said whiskey. Someone said, "Oh, Freudian slip," because I said whiskey. What? And it's like, you need some whiskey. Yeah, okay. But no, whiskey could lower ADH levels. Okay. So, or you can just drink a whole bunch of water. When are you guys doing the lab in Physio 2 where they're making you drink the stuff? This week? Okay. So, okay, what groups did they give you guys? That you had, someone's drinking distilled water, someone's drinking, which salt? There's a 2%, there's a 0.9%, is it 3%? Is anybody doing the bicarb? No. It's very, the bicarb is not fun, but it actually has cool results, but it's very unpleasant. The high percent saline is really not that bad. Trust me, if I gave you the bicarb, you'd say, give me the saline, okay? So, if I drank the really high percent saline, what's going to happen to ADH? I drink like 3% saline. It's like injecting me with 2% or 3% saline. What's going to happen to the osmolarity of your blood when you drink a very high osmolarity solution? It's going to go up, and what's that going to do to your urine volume? It's going to go down. So those of you who drink that, you're going to notice your urine volume is going to actually go down. Okay? If you drink the distilled water, the osmolarity is going to go down, and your urine volume is going to go, what's it going to do? Is it going to go up? Yes. Wait. Yeah. Okay. Think about it. So you're going to you get to actually measure that. Okay. If I gave you 0.9% saline, what's going to happen? Yeah, you're going to pee it out, and it's like it's not going to dehydrate you. It's okay. It's not so bad. But changing the osmolarity of the solutions give you can change what's going on. Okay. So let's look at our first scenario, and I'm using my limited knowledge of remembrance of Spanish I took in high school. I did take Spanish in high school. That was actually pretty good, but it's been years and years and years, so I don't remember it anymore. Muy importante. What does that mean? It's very important. Okay, so what's very important? So if I want to excrete a dilute urine, urine is going to be what relative to the plasma? Well, you can say dilute or concentrated and then use the fancy term. So if you want to, so you already said dilute. If you want to excrete a dilute urine, okay, urine is going to be what in regards to plasma? Hypo, hyper, or isoosmotic to your plasma. If you're excreting a dilute urine, it's going to be hypo, okay? It's going to be hypo. So what's so important in order for this to happen is, you remember, you need to be able to separate water reabsorption and solutes. You have to, some parts of the tubule, you got to have to have water coming in or not. So actually with this one, I want to reabsorb sodium, but I don't want water to follow, because otherwise you're not going to be able to excrete a dilute urine. So certain parts of the nephron tubule, you're going to reabsorb sodium, but water's not going to follow. And so you want, automatically you have parts of the nephron tubule that are always impermeable to water. And then the other ones, it's going to be impermeable as long as you don't have ADH around. Okay? So let's pull out our little nephron and go through what's going on there. But even though technically in this discussion, I really only care about the sodium in the water. But just for grins, I'm not going to draw it on here, but I'll just ask you what's going on in the different parts of the nephron tubule in regards to like glucose and other things, just to review. Okay? So we're really only concentrating now more about sodium and chloride in water. All right. So we've got this structure right here. What's that structure? It actually doesn't include that. It's the glomerulus and the little Bowman's capsule. What do we refer to that as? The renal corpuscle. And what process occurs there? Filtration. And remind me what gets filtered, or actually it's easier, what doesn't get filtered? Blood cells and large proteins. But other stuff gets filtered. Okay? So you have all this stuff being filtered. Okay? Glucose, sodium, chloride, potassium, bicarb, blah, 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 blah. Okay? All this stuff gets filtered. Waste products too. What's the first part of the nephron tubule referred to as? Proximal tubule. Okay? So since we're talking mainly about regulating volumes, I'm really going to emphasize what's going on with sodium because it kind of drives everything. But sodium, what's going to happen to sodium in this portion of the nephron tubule? Will it be secreted or will it be reabsorbed? I heard it's secreted. So how many for reabsorb? How many for secreted? It's reabsorbed. I want to get it back. So you're going to reabsorb it. What mechanism are we going to do it? Is it passively or active? It's active. Okay? So remember the sodium-potassium pump. It's creating this favorable gradient allow sodium to come in. Sodium potassium ATPase uses its primary active transport. You're going to direct expenditure of energy. It creates that favorable gradient. Sodium's going to want to come in from the lumen across the cell, and we have a lot of transport proteins that bring sodium with a whole bunch of other stuff. So sodium, I'm going to put sodium coming in, but who else tends to come in with sodium? It has you have transporters that kind of hitch a ride with sodium. Glucose. Okay, so you got sodium glucose transporters. You got sodium amino acid transporters. You got sodium phosphate transporters. So sodium kind of drives a whole bunch of stuff. Okay, so you're going to reabsorb a mess of things. Okay, do you reabsorb urea here? Is the proximal tubule permeable to urea? It is. Good percent is actually reabsorbed, about 40-something%. It is. Okay, so it is, but don't worry, we'll get rid of it at some point. So it actually is reabsorbed. Okay, so urea is, okay?
So all these are reabsorbed?
Yep. So far, a lot of stuff's being reabsorbed. Now, what's this one thing I do want you to remember that is secreted? No, glucose is reabsorbed. Hydrogen ions. So I'm actually going to draw that in there. So hydrogen ions are actually, I'm going to draw the arrow going that way. And believe it or not, you use a sodium-hydrogen exchanger. Sodium comes in exchange for hydrogen ions. So the hydrogen ions are being secreted. But that's going to be important when we talk about reabsorption of bicarb. But hydrogen ions are secreted. A bunch of everything's reabsorbed. Okay? Who comes along after you got sodium coming in, you got potassium coming in, you got chloride, you got a whole bunch of stuff. Who follows along? Water. Water. So water, remember, why is water going? Membrane's permeable to water, and the reason why it's being drawn with the solutes, it always wants to maintain osmotic equilibrium across the membrane. So it's going to follow it. So the osmolarity outside is the same as the inside. So water, remember, always moves passively. So you're going to have water following everything else. All right? Osmolarity. What's the osmolarity of the tubular fluid all along that proximal tubule? If it's permeable water, it's got to be isoosmotic to whatever the interstitial fluid is, and it happens to be the same as your plasma, which is, well, I mean, it's about 300. It's like actually like 290 something or whatever. About 300 milliosmoles per liter. It's the same as your interstitium as well as actually your blood. So it's about 300. So I'm going to write 300 in here. 300 out here. Okay? It's going to be isoosmotic. All right? By the time you make your way down here, what is this part right there? The descending limb of the loop of Henle. What has been completely reabsorbed? There's some things that have been completely. Glucose. Anything else completely reabsorbed? Amino acids. You said that, okay. So amino acids, she's like, amino acids, glucose, those are completely reabsorbed. Now, the other things aren't completely reabsorbed yet. But actually, majority of your reabsorption of things takes place in the proximal tubule. About 60-something%, almost 70% of the water and sodium that you ultimately reabsorb takes place in that proximal tubule. That's why it's so thick, you got a lot of ATP that's expended there because everything's drawn with the sodium. That's why it's an extensive brush border. When you think brush border, and we talk about brush border in regards to the GI tract. You've got a brush border that you have all this stuff, and there's all these transport proteins in there. Very designed for all that reabsorption to take place. Okay? So we reabsorbed some things completely as long as anything's not wrong, unless I'm diabetic, and then I maybe exceeded my transport maximum and things end up in the urine. Okay? But other things, most of it's reabsorbed. Okay? But I still have some things that may need to keep reabsorbing as we go along. So you now enter the descending limb of the loop of Henle. Tell me about that descending limb. What's up? It's permeable to something. Water, but not permeable to sodium or anything else really. So the only thing you really see going on there is what's going on. It's permeable to water, so water can move across. What direction does it go? It goes out. And why would it go out? Because water only moves passively. What's drawing the movement out? I'm not having any sodium leaving. The osmolarity out here is high. And actually, as you go from cortex deeper into the medulla, the osmolarity steadily increases. And so we'll say, well, it can change depending on how hydrated you are, but we're just going to say 1200 here. But if I'm well hydrated, it's not going to be quite as concentrated. But we'll say 1200 down at the tip. So what will be the osmolarity of the tubular fluid contents anywhere along this? It's going to be what relative to the interstitium? It's going to be the same, okay? Because remember, it wants to maintain osmotic equilibrium. So it's going to steadily become more and more concentrated as you move down. So about here at the tip, it's going to be about 1200 milliosmoles per liter, okay? Then we start moving up. This is your thin ascending limb. I said it's kind of boring. So what are we going to have going on here? A little movement of sodium, a little bit of movement of urea, and sodium, it's going to, not all of it, some of the sodium is going to leave passively because there's a gradient. Because as this becomes more and more concentrated, you're going to have a higher concentration of sodium here. So a little bit of sodium will leave. Urea actually goes in, because one of the reasons why this osmolarity is so high, you got a lot of urea out here in the interstitium. It helps to create that higher osmolarity. Okay, so urea, some urea is going to go in. Okay, but don't worry, I'll redeposit it later, or I'll get rid of it. Then here's where very important part of the loop of Henle. Which one is this? Thick ascending limb. Okay. What happens here? Water? Any water movement? Yes, no, maybe? No. But what's happening? We're going to do some reabsorption of some different solutes, like sodium, potassium. Remember we had that fancy co-transporter? But we'll have some other ions being reabsorbed. So you're reabsorbing sodium and potassium, a couple chlorides, but water's not following. So what's happening to the tubular fluid contents is we're ascending. It's decreasing. So that's what they call it, the diluting segment. It's becoming more and more dilute. Okay? So we've got, and we're going to show you a little animation later about what's going on, this whole little thing that's going on right here, which is going to be responsible for creating this gradient that we got. Right now I just said there's a gradient. So we're diluting it out. Then we get to this part. I said, what's the only part that I really cared that you remembered about what's going on in the early distal tubule? The macula densa cells. Macula densa cells are a component of what? Structure that's important in the regulation of GFR. Well, it's a component, well, it's a component with the afferent and efferent arterioles, but it's called the juxtaglomerular apparatus. So remember, in reality, that thing's touching the afferent and efferent arterioles, which were important in the regulation of GFR. Okay? You'll have, in the distal tubule, you're going to have reabsorption of sodium and other substances too. But but there's no permeability to urea. No urea up here. No urea permeability, none right here. Okay? I want to start into the late distal tubule, where it's kind of depending on what's going on. So in the late distal tubule, I'm just going to cut it off here. We have sodium that can be reabsorbed. And we can also get potassium secreted. We can get net potassium secretion by specific cells located in the late distal tubule along with the cortical collecting tubule. What were the names of those cells which are sensitive to aldosterone that allows me to get sodium in, potassium out? The principal cells. So principal cells are involved in hormonally regulated regulation of sodium and potassium. Okay, so those cells, we're going to pull it down, are located here and here. What were the other cell type that I had mentioned? Intercalated. They were more important in acid-base balance. Okay. Now the cells here do have ADH receptors. So whether or not water is going to move depends on whether ADH is present. I want to make a dilute urine. Do I want a lot of ADH around? No. So no to ADH, so I'm not going to draw any water moving. Okay? So you're reabsorbing solutes, but water is not following. Okay? Then we start going into the medullary ducts. You still have some reabsorption of sodium in other solutes there. So we can still have some sodium coming in, but there's no ADH around. So what's going on? No ADH. Is any water moving? No. So you got some liquid here, and it's like letting it evaporate. You got liquid, some salt in it, and you let it sit there and evaporate. What's going to happen? As the liquid is leaving, you're leaving it. What you have left is more and more. Actually, no, we're not doing that. Sorry, that's the opposite. We're having solutes leave. Sorry, just telling it. I flipped my thing. So you have solutes leaving, and the liquid staying. So you have a very dilute urine. So you're going to have a large volume of urine, which is very dilute. Okay? Again, what's the major solute, though? I'm still going to measure some solutes in there. There'll be some in there. Mainly it's going to be urea, a little creatinine. You may have some electrolytes still left in there, but it's going to be a dilute urine. Do you want that? If you just drank 2 gallons of water, do you want to get rid of that excess fluid? Yeah. Okay. So when you want to produce a dilute urine, every step along here, like we did last time, is the same. The only difference is whether or not ADH is present. That's the only difference. Here, ADH isn't present, so we're just going to be reabsorbing solutes, but water stays behind. Okay? So what if I want to make a concentrated urine? Can I speed the whole process up of lecture by saying, what if you want to make a concentrated urine? Is every one of these steps pretty much going to be the same that you see here? The only exception is going to be what? ADH. If ADH is present, what? Water is going to be leaving, but we're going to add one more, and we'll go over it again. The ADH also increases the permeability of the medullary ducts to urea. And so what also happens is more urea leaves, but the urea is going to contribute to the osmolarity here. It's going to draw more and more water. So as you move down here, because it is permeable to water, and you have an increased osmolarity of the interstitial fluid, you can take more and more water with you. And so as you get down here, what's going to be the osmolarity of the contents right here? It's going to be identical to what you got over here, which would be maximum in a human. It's going to be about 1,200 milliosmoles per liter. It's going to be very concentrated. That's good if you don't have ready access to water. So we actually sped things up. It's the same thing. It just you'd messed up what's going on here. But we'll go over it again. Don't worry. Okay? So depending on you want dilute urine or concentrated urine. If I'm exercising, yeah.
Where is it 1,200? I couldn't see.
At the very tip, down at the very lowest part of the medulla, okay, right close to the renal papilla. It's going to be about 1,200 milliosmoles per liter, okay? Now, if I'm exercising, what's happening to fluid loss when I'm exercising? Am I going to be losing some fluids when I exercise? Some things that are inevitable. So you're sweating to cool off. Breathing. Now, what breathing? What's that an example of? Insensible water loss. So you're going to increase that, but sweating is not insensible. But we're losing water. Maybe if I don't have ready access to water, how am I going to be able to try to maintain water balance? What happens to urine output? It's going to decrease it. So kidneys are going to play a very important role in trying to maintain water balance by affecting what's going on with urine volumes. But urine volumes can be affected by changes in plasma osmolarity, depending on what's going on. So we can tie it back to an earlier lecture when we said, hey, let's eat a whole bunch of salt and see what happens. And what's going to happen to in extra, I can tie it in what can go on to urine volume. I can do it. You want me to do it now? I'll do it. Because I'll tell you what we're going to talk about later. If I eat a whole bunch of salt, yum, yum, num, 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 okay? What happens to your osmolarity of your blood if you're eating a whole bunch of salt? It goes up. Plasma osmolarity is regulated by the hormone ADH. And that's something we're going to be talking about at the end. ADH, do you want more ADH or less ADH if your osmolarity is high? I want to dilute that thing out. So you want to increase ADH so you can hold on to more water and actually dilute it. But here's the problem. You hold on to water, what happens to your blood volume? You got more water in there, it goes up, and what happens to your blood pressure? It goes up. But kidneys kind of compensate for that, and what happens to your urine volume? Now, if your blood pressure goes up, it actually increases. Your kidneys are going to try to compensate for it, but as long as you're consuming all that salt, it's going to keep going. So your blood pressure is going to stay elevated until you stop eating all that salt. So high sodium diets could influence blood pressure, but other things can influence blood pressure. So while I remember, say, hey, I got high blood pressure. Can I target the nephron tubule as a means to regulate my blood pressure? Yeah, and a lot of people are on diuretics. In different parts, there's different drugs for different parts of the nephron tubule. And actually, there's one that loves to target this one. So what happens if I inhibit that pump right there? What's going to happen to my ability to concentrate my urine because I've blocked this pump? And I'm going to tell you right now, this pump is very important in creating this gradient that we see here. I block it. What do you want? Just think logically. If I have high blood pressure, what do I want to do to get my blood pressure back down by using a diuretic? I'm going to pee, and if I pee, I can help decrease blood volume to decrease blood pressure. So a lot of the diuretics increase urine volume, but they target different parts of the nephron tubule. So there's a specific one that targets this. There's some that target other places too. Okay? So they're used to decrease or increase urine output to decrease blood pressure. Okay? So let's go back and talk about something before we look at if I want to make a concentrated urine. Oh, here we go. There's something called obligatory urine volume. You're obligated. That means you got it. You're supposed to. You have to do this. You have to have a certain amount of urine that you excrete, a certain amount of fluid. Yeah.
So, like with drinking like seawater, that's what kills you, right? So initially, I'm taking all the salt and I'm going to hold on to the water, but then with high blood pressure, then I'm going to get out. Is that?
Well, if you drink salt water, what happens when you drink salt water? And so it goes back to that one lecture we talking about osmolarity. So if I drink salt water, what happens to the fluid inside your cells? Gets drawn out, and it's actually caused what we call more of like a dehydration in regards to intracellular. You can have, you ever talk about edema? Edema could be extracellular, intercellular. Same way with like we're talking about loss of fluids. If you lose water from your cells, that can cause dysfunction there. So it's dehydrating you in regards to it's removing water from the cells, but the cells won't function properly. Okay? So, but yeah, they'll kill you and mess up your kidneys too. So yeah, don't drink salt water. Not a good thing. Okay? Well, I mean, you guys are drinking a little bit. That's enough. Just short term. Okay? But you're not all dehydrated when you drink. I was going to say, me, I don't want to drink salt water now before you go to Latin. It's not good.
But that's from the interstitial, not because of in the long run being out, like due to the increased salt, like we were talking about. It's not because of that, right?
Well, no, because if you drink salt water, you're like, well, ADH levels should go up, right? And okay, now short term, like if you're all hopefully are normal. Okay, we're hoping. Okay, so and this is why it's not going to kill you. Okay, you drink some salt water. It's going to draw some fluid out of the cells into the extracellular space, but ADH levels are going to go up because you're drinking a hyperosmotic or hypertonic solution. What is ADH going to do? It's going to allow you to hold on to water to try to dilute it, and then at some point, then we're going to eventually normalize it, and we're going to pee it out at some point. But hopefully everything's good, as long as you're not dehydrated and you're drinking a whole bunch of salt water. Okay. All right. So you're obligated to have a certain volume of urine because you got to excrete waste products. We are not birds. Birds, the uric acid, you know how it's white and it's not liquid. I mean, it's kind of liquidy. But we have to have urea needs to be in a certain amount of water to get rid of. So we're obligated to get rid of some waste products. Okay? Now, do you know the difference between fish and humans in regards to their nitrogenous waste product that they excrete? A little comparative physiology, anatomy. What do the fish excrete? Which is actually more toxic than urea. What did we make urea from? What did the liver convert to get urea? Well, it's from amino acids, but then the amino acids were metabolized and you made really toxic. The amino group, you can make ammonia, and then you converted ammonia to urea, which is less toxic. Fish pee out ammonia. Is that a problem? Ammonia is very toxic. Is it a problem for the fish? As long as it's not in a fish tank that someone didn't been filtering and cleaning. If I'm out in the ocean, is it a problem? I'm surrounded by a whole bunch of water and it's washing it away. So fish can get away with ammonia. We have to get away with urea. It's a little less toxic, but we still have to excrete some volume, not as much as the fish, to be able to get it away. So we have to excrete these waste products. So we're obligated to get rid of a certain volume of water. And so how you can figure out what's your obligatory urine volume is you're like, well, how much waste products do I make? What's the maximum concentration of urine that I can get? And it's a little bit of math. Humans, max is about 1,200 milliosmoles per liter. And we're producing about that much of waste products. So this is what our minimum urine volume should be, a half a liter. So someone who's very dehydrated, the minimum urine volume is about a half a liter per day. If you're very well hydrated, will that volume change? Do we get rid of more? We're getting rid of the same waste products, we're just getting rid of excess water. But you're obligated, humans are obligated about 500 mils per day. What about fish or animals that live around water versus animals that live in the desert? Do you think their obligatory urine volumes may be different than a human? Okay, so think about the beaver, the lonely beaver, unless you're from Duck Dynasty and the beavers are your enemy. Okay, you know what I'm talking about? Damn beavers, because they do a lot of damage. They have to stop. Okay, they're always building their dams. Okay, so beaver hangs out around some water. It's building those dams because it wants to hang out in some water. Do you think their obligatory urine volume is higher or less than a human? They're hanging around water. Yeah. No, actually, you sure? It's actually higher because they have more ready access to water than we do. So that, remember, so how they can get away. Their urine volume is actually a little higher. They can't concentrate their urine as much, but they're right by the water. They're in water all the time. We aren't floating around in water all the time. What about an animal who hangs out in the desert, who doesn't have ready access to water? Do you think their obligatory urine volume is higher or lower than ours? Better be lower. But how can they do that? Can they concentrate their urine more than us? Yes. So it all comes down to how much you can concentrate your urine. So the desert dweller, animal, certain animals, and I'm not talking about me, humans out in the desert. No, they're still going to be same as regular humans, okay? An animal that's evolved, that's been living in the desert, that doesn't have ready access to water, what will be their maximum urine capacity, concentration capacity? Is it going to be higher or less than the human to be able to concentrate their urine? Higher. Will it be higher than the beaver? Yes. So it'll be the desert-dwelling animal, the human, and then the beaver, or any other animal you choose that hangs around some water. Okay? So let's just look at my beaver, and this is what we call an Australian hopping mouse. Okay? The Australian hopping mouse lives out in the desert there. Okay? So look at how concentrated its urine could be. 10,000 milliosmoles per liter. That's pretty concentrated. The beaver, it's less than us, about 500 milliosmoles per liter. Okay? That's kind of good to evolve that. One of the reasons, too, that it's able to concentrate their urine so much is its loops of Henle are very long. So remember, I said ours is about a maximum concentratability is 1,200 milliosmoles per liter. Wasn't that what we got at the tip, down at the very end of the medulla? But if I got larger space, can I create a better gradient? And I can create a more concentrated urine? They got big loops, long loops of Henle, and they can concentrate their urine much greater than ours. So it's good if they want to live out in the desert. If they couldn't, they wouldn't be. It's like Darwin's survival of the fittest. You haven't evolved that, you're going to die, and you're not going to persist. These things evolved, and they hang out in the desert. Okay? So if I want to make a concentrated urine, there's a lot of things that need to be in play. Okay? Got to have ADH around. Why? Why does ADH have to be available? What's the whole purpose of ADH? Allows you to do what? Hold on to the water. Increases water permeability in the certain parts of the nephron tubule. So water leaves and the solutes stay, or what's left gets more and more concentrated. So you better have ADH. Okay? So if I have diabetes insipidus, should I go be out in the desert if I have diabetes insipidus? No, because what's my issue if I got diabetes insipidus? I either don't make ADH or I don't respond to it. So you better not if you have diabetes insipidus, you better be drinking and drinking and drinking the water. And you better not be out in the desert. You're not going to make it. Okay? Because remember, they have very large amounts of urine because they lack ADH. Okay, the renal medulla needs to be hyperosmotic. That's what I kept saying. So you go from cortex down, you have this gradient. It becomes more and more concentrated, so it draws the water out. Because remember, water only moves passively. So it's more water will be drawn out, the more concentrated its surroundings is. Because remember, it wants to make sure the osmolarity is the same across the membranes. So you have to have a hyperosmotic renal medulla. Okay? And in lab, didn't you play with the PhysioX experiment and mess with the concentrations? Did you alter the concentrations and you see how much urine volumes that you had in response to it? It's true. Okay? We're going to look at an animation when you get back from the restroom break that goes over what you see here, this countercurrent multiplier. It all comes down to that loop of Henle. The loop of Henle is going to be what's responsible for what we call countercurrent multiplication. that will create that hyperosmotic gradient. So that loop of Henle better be working. And how does that loop of Henle, why do they call it countercurrent? Because what? The descending limb is like this, and the ascending limb is like that, running countercurrent fashion. So we're going to look at an animation to show you how it all gets set up. So you have to have this countercurrent multiplication, allows you to be able to have, concentrate your urine under certain conditions, or have a dilute urine under certain conditions. Countercurrent exchange, this comes down to the vasorecta. What's the vasorecta? Who remembers when we talked about vasorecta before? What are vasorecta? Vasa. They're something like vessel. They're capillaries. So they're capillaries that you're like, well, they're around the tubules, but we don't call them peritubular capillaries. Those are associated with which nephrons? They're very important in water balance. The juxtamedullary. So those are the ones that the loops of Henle go deep down in the medulla. Most of the nephrons in your kidney, though, are cortical. Only a little bit of the loop gets in there. But the juxtamedullary nephrons, those ones, the loops of Henle, go deep down in the medulla. The vasorecta are the vessels that are surrounding that. So they're going to be important in that they're giving blood to the tissues in that area, but it's designed in a way that it's giving the blood and the nutrients, and it's not washing away all those sodium and the urea that you just created into the interstitial fluid. So it's not going to wash it away. Now, the vasorecta run countercurrent too. The blood vessels run countercurrent to each other as well as to the nephron tubule. So if I have a descending limb, vasorecta is actually going opposite direction. So we'll show you a little picture a little bit later. So vasorecta is going to be important in maintaining the gradient. Who's important in creating the gradient? The countercurrent multiplier, which would be the loop of Henle. Okay? Then urea is part of the osmotic, creates the osmotic gradient. So urea is that breakdown product of protein metabolism, and that's going to be involved in creating some of that osmotic gradient. You got sodium in there, you got a little chloride, you got some urea. Okay? So all those things need to be working right in order to have a concentrated urine. Okay? So, good time to take a break. When you get back, we'll go through that. We'll go look at the animation. All right, let's watch an animation that shows you what is going to create that osmotic gradient in the interstitium. Okay? If I want a maximum concentrated urine, I want that gradient to be as high as I possibly can. If I'm well hydrated, I don't need it quite as much. But I want it as concentrated as I possibly can. So we're going to see, like, how is this all set up? And what this is trying to depict is what's going on in the loop, and that loop of Henle. So let's kind of see how it's
Functional distinctions between the
occurs.
descending and ascending limbs of the loops of Henle of juxtamedullary nephrons are critical to the establishment of the vertical osmotic gradient in the renal medulla. The descending limb is highly permeable to water and is the only segment of the tubule that does not actively extrude sodium chloride. The ascending limb actively transports sodium chloride out of the tubule and into the interstitial fluid. It is always impermeable to water, so salt leaves without water osmotically following along. Even though the flow of fluid is continuous through the loop of Henle, we will visualize what happens step by step. Before the gradient is established, the tubular fluid and medullary interstitial fluid are isotonic at 300 milliosmoles per liter. The active salt pump in the ascending limb transports sodium chloride out of the lumen until the surrounding interstitial fluid is 200 milliosmoles per liter more concentrated than the tubular fluid in this limb. Water moves passively out of the descending limb until the osmolarities of the fluid in the descending limb and the interstitial fluid become equal. We now advance the entire column of fluid in the loop of Henle. The ascending pump again transports sodium chloride out while water passively leaves the descending limb until a 200 milliosmol per liter difference is re-established between the ascending limb and both the interstitial fluid and the ascending limb at each horizontal level. As the tubular fluid advances still further, the gradient is disrupted once again at all levels. Again, active extrusion of sodium chloride from the ascending limb coupled with the net diffusion of water out of the descending limb, reestablishes the 200 milliosmoles per liter gradient at all levels. As this process continues, the fluid in the descending limb becomes increasingly more hypertonic until it reaches a maximum concentration of 1,200 milliosmoles per liter at the bottom of the loop. Because the interstitial fluid always achieves equilibrium with the descending limb, a gradient is likewise established in the medullary interstitial fluid. In contrast, the concentration of the tubular fluid in the ascending limb progressively decreases as salt is pumped out and water is unable to follow. Note that even though the ascending limb pump can generate a gradient of only 200 milliosmoles per liter at each horizontal level, this effect is multiplied into a large vertical gradient because of the countercurrent flow within the loop. This concentrating mechanism is known as countercurrent multiplication.
Okay, so you notice that the descending limb was bringing all the salt to the ascending limb. The ascending limb was pumping it out. And why was the concentration of the sodium and the chloride so high here? Because this osmotic gradient here is drawing the water out. And so you couldn't this would not happen if these guys were both freely permeable to water. So one is freely permeable, the other one isn't in the way they're set up, that countercurrent fashion. So they have, and I don't know why it's only a 200 milliosmol per liter gradient, but it all multiplies up. So every step, you see it steadily, actually let's do it down. As you move down, it becomes more and more concentrated. So this loop better be working pretty well. That loop is so important in creating that osmotic gradient, which will be important if you want to make a concentrated urine, because in the medullary duct, the water is going to be drawn to all that high osmotic gradient that you have out in the interstitium. So a good target, well, I'm going to say good, we all differ on this one, for people for treatment of high blood pressure is loop diuretics, things that target the loop, because that loop is so important in that countercurrent multiplication thing. So they're not going to have a concentrated urine, they're going to have a dilute urine to get rid of excess water to try to get the blood pressure down. So loop diuretics are target for it. Okay? So one thing though they were talking about or they had the little picture of the sodium chloride leaving. Technically, who also is leaving in that ascending limb? Because you have sodium and chloride, and who else? Potassium. It's actually 1 sodium, 1 potassium, and 2 chloride. It's that little co-transport, but they're just talking about the sodium and chloride, but potassium's leaving too. Okay? All right, so close that out. Fast. They've changed this thing so I can't do it like I used to. All right, so to make that concentrated urine, just review with me. Tell me, we'll just make it easy. Permeable to salt, yes or no, and I'll say permeable to water, permeable to urea. Okay, those are the 3 major players. Proximal tubule, permeable to what? Water, salt, and urea. What direction are we going? Reabsorption. Okay, good. What's the osmolarity of the tubular fluid contents? About 300 milliosmoles per liter. Okay. Descending limb of the loop of Henle. Permeable to what? Just water. What direction is it going? Out. So what's happening to the tubular fluid contents? They're becoming more and more concentrated. Okay, so it's pretty concentrated when you come down here. Then we start going up here. Let's skip the boring part. Let's do the thick ascending limb. So what is it permeable to? Sodium. But remember, we're just doing water, urea, and sodium right now. We've ignored everything else. Permeable to water? Yeah? No? No. Sodium? Yes. Urea? No. Okay. So what's happening to the concentration of the contents in here? Because sodium is being removed, but water's staying behind. It's going down. It's becoming dilute. Okay. Then I move up into the distal tubule, or late distal tubule. Late distal tubule, cortical collecting tubule. Now we're making the concentrated urine. Permeable to sodium? Yes, still have that going on. Permeable to water? Because we're making a concentrated urine. Well, I'm making a concentrated urine. I already gave you that information, so you had to figure out was ADH there or not. So you want ADH around. Okay, so ADH is around. So I said we're making a concentrated urine, so ADH is around. So it is permeable to water. It's not permeable to urea, though. Urea is still staying in there. Then we get same thing going on in the cortical collecting tubule. Then we go to the medullary duct. Permeable to water in the medullary duct? Making concentrate? Yes, because we got ADH around. Permeable to sodium? Yeah, you're still going to get reabsorption. You still get reabsorption of sodium. Okay, so we still have active reabsorption of sodium. So it's permeable to sodium, water. What about urea? Yes. And here's the caveat. I remind you, is ADH will also increase in the medullary duct the permeability to urea. The medullary duct is somewhat permeable to urea because even more permeable in the presence of ADH. Now think about why. Is as the tubular fluid is moving down, water is being reabsorbed. And why is it being reabsorbed? Because the osmolarity is steadily increasing. And I said part of what's responsible for that is urea in there. And so I'll actually be able to deposit more urea there to allow me to be able to make that more maximally concentrated urine. So it actually increases your urea permeability also. Now, is urea movement across membranes active or passive? It is. It's passive. It's always passive. So you're like, crap. So that means urea has to go from high concentration to low, and you got to be permeable to it. So it's like we got a certain osmolarity in this thing. It's got to be presence of some solutes. What do you think is the major solute sitting in there as we move through the tubule? It's urea. So still, I mean, as long as the content's in here greater than here, it's going to leave. Okay, so it's got to remember, it always has to be passive. Urea can only move passively, and you got to be permeable to it. Anywhere. It's always, urea is always passive. We don't have any kind of active transporter that allows us to move urea. It's just completely passive. As long as the membrane's permeable to it, it's going to go from high to low in order to try to maintain generally equilibrium. Remember, that's generally everything's trying to reach equilibrium. Okay? So, we did that. Urea is going to be, already just talked about that, good part of the osmolarity and medullary interstitium. Here's a little thing. What do we get urea from? It's a waste product. What do I need to eat to eventually get urea formed? Protein. Okay, so it's one of the things that in how you metabolize proteins, and it's a waste product, and it's less toxic, I said, than ammonia. That's how the liver comes into play. Now, what happens if I have a very low protein intake? Could that affect my ability to maximally concentrate my urine? If Dr. Case is asking you, it's probably yes. She's even bringing it up. It does, but why? If I have a low protein intake, I'm not metabolizing as many amino acids, I'm not producing as much urea, and your maximum concentrating ability goes down. So you're not going to be able to concentrate your urine as much. You know what also happens, though? Oh, that's a different story. Never mind. I'm not going to change it. I just went in my head, but I forgot. No, I didn't forget. I was like, no, I'm not going to go there. But if you have a very low protein intake, that can impair your ability to concentrate their urine. So low protein intake being lost in the desert, well, if I'm lost in the desert, I'm probably like having a hard time getting water and food unless I'm really good at killing the little reptiles that are out there. Kill the reptiles. You need the meat. Okay? Eat the protein. And we were talking during break about being lost out in the ocean. It's like if you're, and I saw those survival shows. You're lost out in the ocean. It's like if you have a thing that can convert salt water to fresh water, it's a plus, but not. But if you go and try to kill some fish, you can get some water from just eating animals, but suck the eyeballs. Yep. There's water in the humor of the eyes. You can suck the eyeballs. I saw them do it on TV. Some of the ones I'm like, but you will do it if you have, won't you? Just like die, you'll eat your neighbor if you have to. Okay, if you're going to survive. I might say it's a different story if it's a family member. You're like, maybe or maybe not. Okay.
Is that
So.
Naked and Afraid?
Huh?
Is that Naked and Afraid?
Well,
Have you seen this film?
yes, I have seen Naked and Afraid. But the one the one with the eyeball thing, it was I think it was the guy and his wife. Not Dual Survivor, but there's this one with a guy who took his wife out, would do the survival thing with his wife. I can't remember. But there's so many different ones out there, but not Naked and I only saw a couple episodes of Naked and Afraid. I can only see so much blurred images after a while. Yeah. It's like, I don't know how they can do that. That would just be so uncomfortable to walk around naked like that all the time. But they always, do you notice they always they devise little outfits after a while? The girls somehow manage to make some sort of outfit. You notice that for those of you who've seen it? Do you know what I mean when he said Naked and Afraid? They throw a man and a woman out completely naked, strangers, and say, hey, survive. You say, it's like, really? You say, awesome, ain't it? Actually, a couple I've seen, the girls were better. The guys were like, suck it up.
The guys were like, I can't do that. And the girls were like full-blown survival instructors.
I know. The guys were like, eh. All right. Oh, okay. Changing gears. If there's excess water in the body, tell me. You got excess water, ready access to water. How much urine are you going to make? More or less? You got excess water. More. Get rid of it. Okay. Is it going to be concentrated or dilute? Dilute. Okay. Urea movement, diffusion of urea in the medullary interstitial fluid. Am I going to have more urea going in the interstitial fluid or not? Less. Don't need it. I don't really need to concentrate it. Okay. Less. Okay. ADH levels, high, low. Low. Okay. Permeability of the ducts to water and urea. Low. Okay. So we went from talking about dilute urine, concentrated urine, back to dilute urine. Okay. So, okay. What about the vasorecta? So we created that gradient because of that loop of Henle, the countercurrent multiplier. What's the role of vasorecta? That's the countercurrent exchanger. So these are blood vessels. They're kind of around that loops of Henle, and they're there to give some oxygen, nutrients, pick up some waste products. But here, think about it. If I'm a blood vessel, and blood vessels are capillaries are permeable to a lot of stuff. I'm moving on down. Ooh, there's tons of salt right here. Where does salt want to go? If it's permeable, it's going to be permeable to sodium. Where's the sodium want to go? I'm a blood vessel going down. Ooh, there's a lot of sodium right here. Where does the sodium want to go? Right into me. Now, if I'm a blood vessel and I'm going down into that medulla, which has a high sodium concentration, urea concentration, sodium and urea is going to go in me, and I kept on going, guess what? You've pretty much removed your gradient. You've just defeated the whole purpose. It was like, I'm not going to be able to create a gradient if the blood vessels are arranged a certain way. But the vasorecta are arranged countercurrent. So here we'll say you guys are the descending limb of the loop of Henle. You guys are the ascending limb of the loop of Henle. And I'm going to be following the direction of blood flow from the vasorecta. Okay? So I'm moving on down. Actually, I'm going to do this way. Go this way. So you're the descending limb. Okay? So I'm the vasorecta coming up by you guys. Oh, guess what? I'm going to take out some sodium. Say, hey, there's all this sodium here. So what am I going to do? I'm going to turn around, though, and I'm going to deposit it back. So if I just was a blood vessel that just went in one direction, I can take away all that sodium. But if I'm arranged in a certain way, I'm going to bring in what I have. I'm going to pick up some sodium, but I'm going to redeposit it later as I come back up. So it's nice that they're arranged in a countercurrent fashion. So the loop of Henle's arranged countercurrent, and the vasorecta is arranged countercurrent. So it's just enough blood flow to keep you happy, but not too much. You don't want a really fast flow rate either, do you? Do you want a lot of flow rate, things going a little bit too quickly, so you might not have time for, if you can actually wash away the gradient?
Why is the salt going in? Why is the fluid that's in the vasorecta not following the solute and going out, actually? I guess I think about that.
Here, let's do that. Here, let's see if we can do it in picture form, maybe. See if I can do it in picture form. All right, so here's my vasorecta. Here is my loop of Henle. So you notice that the vasorecta run countercurrent to itself as well as to the loop of Henle. Okay? So here's the vasorecta running down where you have a lot of sodium. The sodium is increasing. So this is permeable to sodium. So doesn't sodium want to come in to the vessel as we're moving down? Sodium's coming here, sodium's coming here. Would you agree? Because it's highly concentrated. As it moves up, what's going to happen is the sodium is actually going to be redeposited as it goes up, because also what's happening is you got to have stuff going on with water too. So let's see if we can figure this out. What's going on with water in the descending limb? Where's water going? Going out. Where's it going? Well, interstitium, but then also who? Where's it going in here? The blood vessels. So it's going in the blood vessels. And so what's happening is it goes into the blood vessels. The water's it goes from here and into the blood vessels and then on up. But pretty much, try to put it in a nutshell.
Is it the gradient? Is it the sodium gradient? Because it just seems that why would the salt go in if the water follows the solute, not the solute follows the, you know what I mean?
Well,
Or
Well, here, the reason why water is leaving here, because it's permeable, because it's following where all the solute is. But you're like, well, then shouldn't it come in here? I mean, into...
I guess probably that's my question. Why doesn't the
But
water
oh, here,
flow into
you're saying, why isn't it? Because the water actually wants to also leave here because it's drawn to the sodium in here.
And so the sodium going back and forth between the interstitial and the peritoneal, the
Vasorecta.
Is that just the gradient that's driving that factor? Is that what that is then?
No, with the say it
On,
another way.
like on the ascending side, is a high gradient because it's being pumped out from the ascending, so it's trying to go into low gradient of the vasorecta, and then on the opposite side, it's the exact opposite, right? Because of the salt not leaving the descending.
Yeah. Mm-hmm.
So that is it the gradient that's driving that forward?
Yeah, and actually what's going here is, we'll probably put it in a nutshell, is whatever's being just enough that's picked up is deposited back. It ends up being kind of equal. By the time this blood vessel goes up here, it's back to where it was. It's like 300 milliosmoles per liter. If I was going to kind of sample the blood down here, it's going to be pretty concentrated. But by the time I get up here, it's going to be back to where it needs. So what comes in eventually is going to come out. The reason why you can do it is the way it's arranged. Is only because of the way it's arranged that you can do that. So it's just pretty much just bringing enough blood to keep it happy, but it's not going to break down that gradient. Okay? But if you have a lot of flow rate there, it would break down the gradient. Okay? All right. So let's do a little clinical applications. First one I already talked about. Kangaroo rat, actually call it the Australian hopping mouse. There's a kangaroo rat too. I guess where the kangaroo rat lives. Where's the kangaroo rat? Probably Australia and some other deserts. Okay. Why does the kangaroo rat or the Australian hopping mouse have a longer loop of Henle than the beaver or even me? It's a hot environment, arid environment, doesn't have ready access to water, so what does it need to do? Concentrate its urine more, and the only way you can do that is if you remember that countercurrent multiplication, you got to have pretty long loop of Henle. If you have longer loop of Henle, the greater the gradient, so they can concentrate their urine better. Okay? And I want to know who actually measured that, but you know, someone did. Okay? Who took the urine from the Australian hopping mouse? I don't know. Was it me? Okay? Ever have your specific gravity or urine measured? And that's actually one of the things you'll be measuring in lab after you drink your liquid. Okay? So specific gravity, it's an indirect measure, which is the reason why we're doing it in lab, is it can give you an idea about a subject's hydration status. Literally, though, it's a measurement of like a weight of a substance in that fluid. But if I'm dehydrated, what's going to happen to my specific gravity? I still have the same amount of solutes, but my fluid volume is changed. What's going to happen to your specific gravity if you're dehydrated? It's going to increase. So we're going to use, that's not the only thing that can affect specific gravity, but we're going to use that as an indirect way of looking at your hydration status. If you have ready access to water, your specific gravity is going to go down. But remember, that's not the only thing that can affect specific gravity. So if I have, say, renal disease, damage, and I get a lot of proteins in my urine. Am I supposed to see proteins in my urine? A lot of proteins. I mean, you have a little bit, small ones. But if you start getting a large amount of proteins in your urine, what do you think that'll do to the specific gravity? It's going to increase it. But obviously they're testing it. Like, proteins, what's going on? I need to figure out what's because I'm not supposed to see proteins in my urine. Anything else I'm not supposed to see in my urine? That if I test for, I'm like, ooh, red blood cells. You know, it's like, but the red blood cells could be coming from just the ureters, but it can be coming. You have problems with the glomerulus. White blood cells can indicate you got some sort of infection. Should you see glucose? No. If you see glucose, you start thinking, ooh, we better check for diabetes mellitus. We need to check for that. I don't know if they even test for that. They just test for protein. Yeah, I don't think they measure amino acids. They just test for protein, I think. Maybe they do, and I don't remember. What about ketone bodies? We all have a little bit, but what happens if your ketone body levels are up? What could that indicate? Because that's something that's on the strip. If you have a very low carb diet, you can have a lot of ketone bodies. Or on the Atkins thing, that could do it. If you're diabetes mellitus could cause ketone bodies to go up. Doesn't always, but it can. Okay. So you got like you're looking for certain things. Things you're like, what color is the urine supposed to be? Maybe a very pale yellowish color, pale. Now, if you have a concentrated urine, what does it look like? Because there's not as much fluid there, it looks darker, right? If you got lots of urine, it may be a little bit less light. So you always need to look at the urine. Is it red? Is it green? I mean, your urine can change colors depending on what you eat. Stuff dyes in your food can affect the color in your urine. Long time ago when I was in college, I took these diuretics. Never do that again. It made me sick. But my urine was like a greenish, bluish color. Freak shit, but I knew because I was like, I took the diuretics. But you can change color, so what you eat can do it. It has to be a certain smell. I'm not telling you. When it first comes out, it shouldn't really have much of a smell to it, but it can have a smell. Okay, so it depends. So you're going to look for certain things in it. So, yep. What?
While we're on those applications,
Okay. All right. Oh, okay.
like survival shows, what do you think about like drinking urine in a survival situation?
They say you can only do it like one pass. I think it is like one pass, but because urine is sterile. But I think one time, one pass. But I would, if I could, I would kind of distill my urine. And I did that in one of the survival shows. They put the urine in like a plastic bag, like a little Ziploc bag type thing, with the sun and do the thing, and they're trying to kind of distill it, doing their own distillation type thing.
So physiologically, it's not extremely detrimental, like one time you think?
I think they say one time is not going to hurt you. I'm not saying, hey, let's go try it, but
Right.
if I had to, oh yeah, I'm going to. I will, if I had to. But yeah. But they said it's only like one time you can really do it. Maybe twice, but I can't remember. All right, so we're going to have, I think Thursday, instead of big board game, we should have survival. See who survives. No, okay. Yeah, 2 hours. You're going to drink urine? Yeah. No. No, it's not Survivor where you have to eat the nastiest foods and drink the nastiest stuff, okay? All right. So, last couple topics, because we got to get through this because this stuff is on your exam, okay? But we have, believe it or not, talked about the majority of this before in previous instances, okay? Sodium and plasma osmolality. Sodium is a huge component in plasma osmolality. It's the major cation in the extracellular fluid, would we agree? And a lot of things, things that affect sodium could affect the movement of water. And in lab, didn't they have, it was like a couple weeks ago, I think, is they gave you some scenarios and said if I gave you the sodium concentration, I could calculate or estimate your plasma osmolality, because it's about 2 times your total plasma osmolality. If I have your sodium concentration, it's a little bit more than 2 times. But sodium is a very important factor that if you mess with sodium concentration, it could affect the osmolality and affect the shifting of water into and out of the cells. So it's an important regulator. Okay, so things that affect sodium could affect what's going on with water balance. Okay, so let's talk about the osmoreceptors. I'll link it to something I talk about. You eat a whole bunch of salt. Okay? Osmoreceptors. These are receptors that are sensing changes in the plasma osmolality. You got them up in the hypothalamus. Hypothalamus does a lot of stuff. So remember, like day 1, we talked about hypothalamus making a bunch of hormones. We did. Like a list of them. Some of them are transported to the posterior pituitary. Others are transported to the anterior pituitary and influences the secretion of other hormones. Well, hypothalamus has these things called osmoreceptors. It's also the site where ADH is actually produced. ADH is made in the hypothalamus and then stored in the posterior pituitary. So you have these receptors that sense changes in plasma osmolality. And this is a great time to review what happens where water is going to go in or out of the cell depending on what's going on to the osmolality. So if I eat a whole bunch of salt, dum, 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 my plasma osmolality is going to do what? It's going to go up, okay? So this is my osmoreceptor, just like any other cell. Volume can change, okay? So my osmolarity is up, so I'm going to put all these little dots over here. Where does water want to go? It's going to want to go out. So what's going to happen? The cell is going to shrink. You know what happens? These shrink, their firing rate goes up. Bam, bam. What is it telling? Say, I need to release more antidiuretic hormone because I need to hold on to water to try to dilute all this concentrated stuff in my plasma. So the osmoreceptors, if they shrink, and they would shrink in response to an increase in plasma osmolality, their firing rate goes up, and they increase their secretion of antidiuretic hormone. If your osmolality goes down, the cells are going to swell, and you don't secrete as much of it. That's the primary means or primary regulator of antidiuretic hormone secretion is changes in plasma osmolality, but it's not the only thing. So we'll talk about a couple other things, but that's the primary stimulus for release of ADH is increases in plasma osmolality. So we said you ate a whole bunch of salt, the cells shrunk, you release a lot more ADH, okay? You're going to secrete it, and so what's going to happen? So we'll follow this one. It's kind of a similar thing. So if I have a water deficit, that could also increase osmolality. Would you agree? If I don't have a lot of water around, becoming very dehydrated, would my osmolality go up? Yes. So that would go up. ADH secretion would go up, hypothalamus and neurohypophysis. What's going to happen to plasma ADH levels? Up. Targets the kidney. And what happens to the water permeability in those latter parts of the nephron tubule? Permeability is going to go up. And so what's going to happen to your urine volume and concentration? Your urine volume is going to go down, and it's going to be more concentrated. You're going to try to hold on to as much water as you can. So I say if you're eating a lot of salt, it's just attempt to dilute it out. But also that can happen if you haven't had enough fluids. Okay? Do you know what else would happen? Though you have a thirst center up there in the hypothalamus. The hypothalamus does a lot of stuff. There's a thirst center up there too. Are you going to get thirsty if your plasma osmolality goes up? That's why at the bar they got the salted pretzels and peanuts. Then you get thirsty, and hopefully you buy a beer. But the beer's not going to help you very much. But so it makes you thirsty. Do you think that'd be kind of important to ooh, I need to drink some more fluids? I'm going to say drink more alcohol. Drink more fluids. It also stimulates thirst, so then you increase your water intake too. Now what if I have water excess? Can we just fill all that in and just switch the arrows? Yeah. So changes in plasma osmolality is a pretty important regulator of ADH, which can affect your urine volume and changing your urine concentration. But they're not the only things that influence it. Notice here, you have input to the hypothalamus from other receptors in your body, baroreceptors. What do baroreceptors do? They're pressure receptors. You have receptors in the heart, in the pulmonary vessels, which respond to changes. The cardiopulmonary receptors respond to changes in volume within, say, the pulmonary artery or the right atrium. The baroreceptors are sensing changes in blood pressure. Would blood pressure affect urine output? So just think about why you'd want to do that. So if my blood pressure is low, what do you want to do with your urine output? Do you want to decrease or increase it if my blood pressure is low? Wait, say again? Decrease it. Why? You want to hold on to as much water as you can to try to get your blood volume up, to help get your blood pressure up. So that's one of the things you can do. If my blood pressure is high, one thing I can do is increase the urine output. And so indirectly, changes in blood pressure can also affect ADH release. So think, and also relate to blood volume. Blood volume changes could affect ADH levels. If I'm losing blood, my blood volume goes down. Do you want to increase your urine output or not? It's like, no. So if I'm losing blood volume, do you want ADH high or low? You want it high to hold on to as much volume. So blood volume can affect ADH. Blood pressure can also affect ADH. So it's not always plasma osmolality. Blood volume and blood pressure can also influence it too. So let's look at this chart that you have in your notes. So plasma osmolality, if it's high or low, does it increase ADH? High or low? Would it increase ADH? Yeah, this is the remember, this is the primary regulator. If a decrease in plasma osmolarity would decrease ADH. What happens to blood volume to increase ADH? High or low blood volume? Low. And same with blood pressure. And then high blood volume, high blood pressure would decrease ADH. Let's do some other things that may do it. We already talked about the alcohol. What does alcohol have to do with any kind of with ADH? Drinking alcohol does what? Inhibits ADH. That's why you start peeing a lot. So why do you feel so crappy the next day? Some of you don't get hungover at all. Okay. So part of it has to do with being, part of it is acetaldehyde. It's the toxic effects of trying to metabolize the alcohol, because you got to metabolize alcohol to acetaldehyde and then to acetate, and that first step is faster than the second step. That's part of the reason why you're crappy, and part of it is because you're dehydrated. So that's why you feel awful, unless you just make sure you stay well hydrated. As you get older, you get hungover a little bit easier because you tend not to be as well hydrated. Yeah, and there's other stuff you can do. A little orange juice helps too. Yeah, okay, yeah. Some stuff that helps. Or drink lots of. Who wants to? Every beer I have, drink a glass of water. A beer, no, I'll be in the bathroom even longer than there's anybody. It's like no, but that also helps if you keep drinking the water with it too. Okay, why is if you're nauseous? Being nauseous is a very potent stimulator of antidiuretic hormone. Why do you think that's been designed that way? If you become nauseous, ADH, like, ooh, better release this ADH. Because what is it trying to do? Or is it a little preemptive strike? Because if you're nauseous, there's a good chance you're going to go bleh, and lose fluids. So it's like, ooh, hopefully I can hold on to some fluids. And so it's like a preemptive type strike. So it's like thinking that maybe you're going to throw up. So being nauseous also stimulates ADH. Again, I want to know who did that test. It's like, and how did they make those people nauseous? They had to have made those people nauseous. Like, what am I going to do to make you nauseous? And let's take your blood and see what happens. Or they probably did it with animals. I don't know. So me, just spin me around in something, because I get motion sick real easily. I get nauseous with a drop of a hat. Okay. Gross lab? No, gross lab. No, that doesn't make me sick. Seeing disgusting things, it's just changing body position real quick, like rapid movements, that makes me nauseous. But seeing body parts, yeah, that don't make me nauseous. Don't bother me. All right, so we had mentioned control, or talk about thirst. Could we regulate extracellular fluid volume by changing your thirst? Yes. If I need more volume, let's drink some more water. But thing is, remember, kidneys can't do it all. I mean, if you're lost out in the desert, it's trying to concentrate your urine, but if you don't get access to water, guess what? You're going to die. You know, I mean, it's going to help you to a certain extent, but you still better get some water. I mean, it's going to concentrate it so you don't have to have quite as much water, but you better get some access to water. So I think thirst is going to play a role here. But let's think about these things that could increase thirst or decrease thirst, and they make sense. Believe it or not, they make sense. Think things that you want to conserve water, the ones that we just went through. We went through 3 things and where it's affect ADH or not. They're like, if you want to conserve water, you better want to drink some more. Okay? So, what changes in osmolality is going to stimulate you to want to drink some more? As long as it's not 2% saline. So, if an increase in plasma osmolality. So that's going to increase thirst. What about blood pressure will increase thirst? And blood volume? Decreases it. So I can just change these ones. Those ones kind of link to this whole thing with the ADH levels. Here's a new one. Angiotensin II. What do you think is the relationship between angiotensin II and thirst? What did it do in the kidneys? What are some of the things that angiotensin II does in your kidneys? That doesn't happen in the kidneys, but it does stimulate aldosterone release by the adrenal cortex. In the kidneys, it constricts the efferent arterioles. It stimulates sodium reabsorption in the proximal tubule. And so think about, okay, angiotensin II, why would you want angiotensin II around to like increase aldosterone? Aldosterone allows you to increase sodium. So you got 2 things that allow you to increase sodium. What's going on if you increase sodium? Where else you can probably want to, what's going to come along with it? Water. So under what conditions you probably want more angiotensin II is probably around? Low blood pressure. That was part of your autoregulation thing. If your blood pressure is down, you're going to release more renin. That stimulates more angiotensin II. More angiotensin II, that's going to have effects on the efferent arteriole, increase sodium reabsorption, affect aldosterone. So, high or low angiotensin II stimulate thirst. So same things you said that decrease blood pressure. It's like, oh, angiotensin II will be elevated. So angiotensin II also increases thirst directly in the brain. You have angiotensin II receptors in the brain, in the hypothalamus, that triggers you want to drink more. Okay? And again, water, not necessarily alcohol. Okay? So decrease in angiotensin II would decrease the thirst. So what about the other 2? If your mouth is dry, do you want to increase or decrease? Cotton mouth. Do you want to increase your liquid intake or decrease it? Yeah, so this one would be over here. Now, I start drinking. Ooh, I'm thirsty. Go. Don't you just stop after a certain amount of time after you drink? Because what happens? The stomach stretches out and it triggers a reflex that says, oh, no more drinky. And so gastric distension decreases thirst. That's why sometimes you're like, depending on what you drank, you're like, you can't drink anymore because it's making my stomach all full and I don't want to drink anymore. So gastric distension would decrease thirst. But isn't that protective? So you don't want your stomach to over stretch out, so you got to wait. Drink that fluid, wait for it to move on its way, reabsorb it, and then drink some more. Okay, you can only drink so much at one point in time. Okay? So thirst will play a role in regulation of fluid volume too, but it's not as a potent mechanism because some people like me, I don't get thirsty. I rarely get thirsty, and I know I'm dehydrated half the time. I just don't have drive to drink any water. I just don't. But some people do. I just don't. So my kidneys better work pretty good.
Is there something behind that? Because people always say drink water. I'm like, I don't remember too. I can have a thing of water.
And just sits there.
Yeah.
Yeah, I don't.
Or whatever. It doesn't matter. I'm still like.
I can go a whole day without drinking any water. It just doesn't. I don't know. There's something wrong with us. I don't know. Probably. Thing is, if I drink water, I'm in that bathroom immediately. You have me drinking more water, I'm in the bathroom all the time. So I don't know. I can conserve that water, and then you give me a little extra, I got to go. So if I need to go on a road trip, and my husband won't stop at the toilet, which he's done that before, I'd purposely dehydrate myself because if I drink one, I'm like, got to go. okay, because he won't let me go. If I go on a plane trip, I don't like to use the toilets in the plane. Freaks me out. So I won't drink anything before I go on the plane. Because no, I don't like, and then they're so tiny, okay? So I don't pee on planes if I can help it. All right, so any questions? Who looked at the email I sent this morning? Did you see the new rule? New rule. It's one rule. One rule. It just gives you an extra chance. So it depends. So it's like you come out and you're like your group did okay, and the rest of your team is like, at least you get something out of it. So, but it's more work for me. I got to keep track. So when you come down, you didn't read it, Dallas? Okay. So off the top of my head, I can't give you the number. So when you come down, you know how you come down, you break yourself up into the groups, okay? Remember when you come down is when you sign the roll sheet. So what I'm going to actually do an extra thing is when you sign the roll sheet, say what group number you are. 1, 2, 3, 4, 5, okay? Remember you get 5 questions to answer, and the points go towards your team points, and you got to get so many points to get extra credit points. Well, if your team gets your group, I should say, your particular group does really well, you can get extra points than your team. Okay, so it depends on how many points you earn during your round. So I'm going to keep track of that. I'm going to keep all those papers separate. So you could potentially get more points than the rest of your team because you did better in your group. So I'm going to reward you for doing well in your group. But remember, it all still comes down. It's still socialism. It still is, okay? Where your team, all your groups together make up your team, you still got to earn points for your team to get, the more points you get together, the more extra credit points you get. But at least I'll help you out. But you still got to do, the points, though, is pretty good. I mean, you still got to do pretty well within your particular round.
Like if you're 3 people that go up, get all yours, you get an extra point?
Well, no, it tells you in the email. Depending on what points you get this a half point or you get an extra point. Okay? So I will keep track of it. So it's more work for me. I'm like, So it's very important that you write down what group you're in. Because I'm going to say, okay, this group got this many points, so I can keep track. So you may get different points than anybody else in your team. Okay? But still, even though you're not getting extra points for color or anything, please wear your team color. It just makes it easier for me to keep track of you when you do your team color. Oh, what the heck? It's cold on me. The test is all of renal physiology except the regulation of potassium and acid-base balance. So it's what I just did today. It's the end. So it starts with body fluid volumes. 13 through 16. Yep. God. And remember, this is the last big board game ever. No. Last big board game ever. Ever. Or at least with me, unless someone else does it and this thing's gone. I can't get it out. No mascots, but you can still do it if you want. I'm caught. It's the thing that had it clipped on me and the little, and then it's like, and now I can't get it back through. Try not to hurt you. Well, it's not gonna hurt me. It's gonna hurt the sweater. And this sweater's so old.